So I'm very pleased to introduce Dr. Philip San Miguel, who runs our Genomics Core. And Philip is going to be talking to you today about a technique that was developed by the core called WideSeq, which is the next-gen sequencing technique that the core uses kind of as a replacement for Sanger sequencing that can do quick sequencing for faculty and researchers. So please put your questions in the chat. If you have something that you really want to know the answer to, go ahead and unmute yourself and ask. Otherwise, I will go ahead and monitor the chat and collect questions for the end. So Philip, I will turn it over to you. Go ahead.
Thanks, Natasha. So I'd like to start off with a parable that was related to me by a sales rep from a now defunct Next Generation sequencing instrument company. And she encouraged me to think of sequencing much the same way that one would think of mulch. So the pitch was that her company's sequencer delivered a dump truck of mulch equivalent, in this case, sequence, which is a large amount, but it would be useful for mulching, say, a larger yard. Whereas her competitor's instruments, and her competitor was Illumina, delivered a barge load. So much, one presumes, that it would cover your entire house and prevent you from even getting inside and perhaps even crushing your roof and destroying all your possessions. She didn't actually add that last part. That was just something that occurred to me as she was relating this metaphor. So that seems pertinent here because I would like to introduce, if you haven't heard about it already, our service called WideSeq. Basically, the idea of which is to repackage that barge load of Illumina data, Illumina being a sequencer. And so the way it works is it leverages next generation library sequencing construction, construction and sequencing technology to allow us to reconstruct the sequence of a non-genomic sequence, such as a plasmid, for around $20. And there's some variability in price depending on where you're listening from and whether you're a member of a cancer center or not. So unlike Sanger sequencing, no primers are needed to be supplied, and the sequence that's produced should, if it all goes well, cover your entire plasmid, not just narrow. This is the narrow as opposed to the wide in WideSeq. Narrow band of 400, 800 bases from the priming site used on the plasmid in Sanger sequencing. It also works with limited DNA. As little as 10 nanograms should be sufficient to produce a good WideSeq result. So to actually just to give you a peek ahead of how this would work, if you're interested in it, is you create a request on iLabs using, and then you would either deliver the samples to us in our submission freezer in the lobby of Bindley or ship them to us. And I'll give you some more information about that later. Samples can be submitted anytime Bindley is open, but processing occurs once every 2 weeks on all samples collected prior to a 2 p.m. deadline. And the next deadline is September 20th. Then the results are going to be returned via an email with a website link about a week after the submission deadline. Just so you know a little bit about me, and I can establish my street cred as somebody you might want to listen to on the subject of sequencing. I direct the Genomics Core. I received my PhD in the Purdue Genetics program, actually no longer exists here at Purdue, in the Department of Biological Sciences in 1998 for work in grain genomics, particularly maize. That was in the laboratory of Jeff Bennetson, if any of you guys happen to remember him. And I personally cast more than 100 14 by 17 inch acrylamide gels, performed S35 labeled Sanger sequencing reactions to be run on said gels, which I dried, exposed to photographic film, and manually read. I have also performed reactions for and ran countless Alpha Express slab gel based system automated sequencer runs and ABI sequencing reactions and run them. I either ran them myself or saw them run on the core that I direct on ABI 377s, ABI 3700, and ABI 3730 XL instruments. I established the core in 1999, and the core moved to the Bentley Bioscience Center in 2020. And currently the sequencers that we're running are 2 Illumina MiSeqs. And I'll get a little bit more into the capabilities of those instruments in a bit. So basically, WideSeq has been around for a while. It was basically, the idea came from Clint Chappell, more or less, who, because he requested a service, that we provide a service to sequence entire plasmids via shotgun sequencing of sheared plasmid fragments. Just so happened around that time, I was looking at a certain technology, a kit that they had at Illumina called the Large Construct Kit. And it significantly offered this kind of an inexpensive library construction method, because, I mean, typically you're going to charge, upwards of $100 or maybe even more for library construction for an Illumina library that you're going to run on that type of sequencer. That's just not going to work for somebody that wants, the sequence of their plasmid. So anyway, working with then Genomics Core DNA sequencing manager Paul Parker, we basically came up with a WideSeq technique, more or less the same one that we use today. We also had our then programmer, Rick Westerman, created a WideSeq, which was a semi-automatic processing backend, and I'll mention what sorts of processing we can do for you also later on, a later slide. Then also that year, genomics technician Victoria assumed the WideSeq processing duties, which she continues to do to this day. Actually, this very day, she's preparing a WideSeq run right now, or will be this afternoon. And then in 2016, another programmer, David Jacoby, wrote a front-end WideSeq process informatics kind of pipeline that helps us out, although isn't really anything that you would see. And significantly, this seems kind of a weird thing, but I think it made a big difference. Is basically, we used to always just wait till we had a reasonable number of samples, or what we considered a reasonable number of samples, before starting a WideSeq run. And the result of that was that there was quite a delay in between each run, so people could wait longer than a month to get the results, which was not anything that made people very happy. So we decided to just kind of bite the bullet and say, no, as of 2021, we are going to do a run every other week. And so we've been doing that, and it actually seems to have been well received. So this is what I'm talking about here with the as far as price comparison. These are the barge. Well, if you want to think about the ultimate barge here, that would be the Illumina NovaSeq S4, the mightiest sequencer on Earth. Produces 3 million bases. That's 3 times 10 to the 12th bases per run. That's it, using this S4 300 cycle cassette, which you could send a pool down to the for example, the IU Center for Medical Genomics, and they would sequence it for you. Well, they might, if they liked you, sequence it for you at this price. And it'll produce about 10 billion reads in 10 lanes. This, you kind of get the impression maybe this is an amount of data that could crush your house and destroy all your possessions. But we're not going to talk about that instrument today. The one we process WideSeq on is this MiSeq 500 cycle. So I want to compare that to a normal Sanger sequencing read. We no longer do Sanger sequencing, the Genomics Core here at Purdue, but you can get probably similar prices to what we used to do back in the day, a few years back. And one of the prices that we had was $4.36. That was for cancer center members. You would get one read. It wasn't paired. It's just a single read of a single primer from whatever your template was. And the price you were looking at for that is just $4.36. But if you wanted to compare that for 1,000 reads, how much would 1,000 reads of that cost? It would be like $4,360. Base length 800 amount of sequence, 0.001 megabase, with a price per megabase of $5,450. Okay. How about if you said, well, forget it, that price is too high for me, I would like a lower price. You could get down to a lower price per read. So here's a MiSeq 500 cycle cassette. Say you just said, I'm going to create a library on my plasmid, and I'm going to run it on this MiSeq cassette. We could drive the price down to, for each read pair that you got, you'd be down below $1, 37 cents per 1,000 read pairs. However, you wouldn't be getting just 1,000 read pairs. You'd be getting 5 million, or actually you could get more than that. But generally it's no lower than 5 million. You can get 10 million reads from this cassette, maybe even more if you want to push it, although your data quality starts to suffer somewhat. So if we're talking about the price, though, you have to pay, you're paying almost $2,000 for that. And most people are not willing to, just to know the sequence of the plasmid, are not willing to pay $2,000 for it. So then we're thinking about, like, okay, if this is this barge load of data, how could we, say, parcel it up into something that would be more useful to people? And really, what's the important thing? Would not cost $2,000. Instead, it would cost closer to $20. So for that price, we invented this WideSeq technique, and then you can look at it, and your price is now between these, like, major extremes. And you have to look at this and say, well, if all I'm doing is looking at price per base, then I'm never going to do Sanger sequencing. It can't possibly be worth this price. So, but there's other factors that cause people to use Sanger sequencing. And a lot of times, hey, all they need is that one read. That's good enough for them. But still, to me, it seemed like it, and also obviously the Clint Chapel, seemed like it's worthwhile to have some sort of service where you would be kind of spanning the gap between those two types of services, a full Illumina run and a Sanger sequencing read. And so that's the whole idea behind WideSeq. That's what we're doing here. Okay, so just to give you an idea of what this even means, if you don't happen to know how Illumina sequencers work, I'll give you some brief background in that. So the question you could answer yourself is, what does an Illumina sequencer sequence? And you could say that it sequences a library molecule, but what is that? And in this case, it's going to be some DNA or cDNA in PCR-sized fragments. That's actually important for Illumina. Illumina uses PCR technologies to create the clusters and end up getting sequenced. So you can't just go and sequence something that's like a megabase long directly. You need to first break it into pieces, and then take those pieces and sequence them individually, and then try to reassemble them on the back end. So basically, this is the structure. Basically, you're going to take your insert, those pieces, and then the way to sequence them is to flank them by adapters, which are of a known sequence. And those adapters tend to have this sort of structure. They'll have basically their distal. This is the outermost point. Your insert's going to be over here. And this is the outermost point here, the most distal part of your adapter. And that's going to actually bind to flow cells to the oligos that are on the surface of the flow cell. I'll show you a picture of a flow cell in a little bit here. There's some indexes. I'll explain that later on, but it's actually just a bit of sequence kind of in the middle of your adapter that you could use to, say, identify a given sample out of a bunch of other reads that are on the same flow cell, but from different samples. And then the rest of it could be your sequence priming site. All right, so then this is kind of a picture then of this. So this is one adapter. This would be the other adapter. And this is showing you how you could get paired end reads. So you basically anneal a sequencing primer here, do your sequencing, collect that data. Then at a later point, you anneal a sequencing primer to the opposite strand, sequence and then sequence across. Again, get some data. In this case, the insert is small enough that the sequence would overlap, and you could actually do a pair merge if you wanted downstream of that. So, and then as far as those indexes go, so those, as I mentioned, they're little bits of sequences that are embedded in the adapters yourself. So then that could be a part of just kind of the variability of your adapter, where you would have like a set of them. Say you have like, I mean, you could think of having like a set of 96 or 384 if you want of these adapters that are identical, except for the section here where the index is. When you read that sequence, then, well, if you used, say you had a 96 well plate, you had 96 different samples in each of those wells, then you could use a different index in each one. So everything else is the same as far as the adapters go, except for the index sequence. So you can do single index reads on Illumina library molecules, or you could do dual index if they have both, if both adapters specify indexes. And I won't go into this in any depth, but basically the dual indexing allows you to do some extra tricks that makes it much easier to do the processing of something like WideSeq. Specifically, it allows you to make combinations, so you can do a set of like 8 by 12 primers. So you buy these 8 by 12 primers that represent the different ends of the adapters, and you can make 96 different combinations of those 20 primers. All right, and I mentioned before that that you need this PCR sized chunk to be able to actually sequence it on an Illumina sequencer. So there are many different ways to get a longer piece of DNA into something that's PCRable. And so this is showing you one of them. It's a process called tagmentation, originally patented by a company called Epicenter, which was purchased by Illumina at a later point. But this is just some pictures from their patent showing the general idea. This is showing the fragmentation that could occur if you have this. This is a transposon called TN5. Well, it's a transposase from a transposon called TN5, and it's actually carrying, well, the protein itself can actually latch on to the DNA and create these double-stranded nicks, or single-stranded nicks that are just this far apart. You can see this is a dimer here, and you get these nicks on each strand, and you get basically the strand can break apart there. And critically, in addition to that, this transposon is carrying some sequence with it. So in its native habitat, which is the E. coli genome, TN5 would be carrying the terminal inverted repeats of its own transposon, the transposon that encodes it, called TN5. The very termini of that are terminal inverted repeats. These sequences are here, and they're specified by this MEDS, which is just the matrix N. They call it the matrix primers. And they're just showing, hey, I don't actually need these things to be connected to each other. They could just have two different oligos there. And if I did that, then the transposase will actually just break the DNA there and add a little tag on. And I don't have to just limit myself just to this matrix end. I could stick a little bit of extraneous sequence on the end there too. And then in that case, I'll get an extra tag on the end of it, hence the tagmentation part. Then I could use this as a priming site and add in a subsequent PCR reaction to add the rest of the adapter on. And then I could create an entire library molecule. So it's kind of like a clever way to do a kind of a quick library construction where you don't have to worry about breaking things apart, say with a sonicator or even a nuclease. You can instead just use an enzyme, and it takes care of both adding a DNA tag as well as breaking the DNA. Okay, so this is what it looks like on the surface of the flow cell. There's a diagram of what it would look like on the surface of the flow cell. which again, I'll show you a little picture of that later on, what a flow cell is. But the flow cell is what you plug into the instrument, and that's where the action occurs, where the sequencing occurs. And then the sequencer watches as each base is added and gives you a sequence back. So basically, this is just showing, hey, I could do a read one. This again, you can see the 3 parts of one of the adapters. Here's the insert, 3 parts of the other adapter. And the other adapter is like attached to the surface of the flow cell in a process I'll go into just a little bit in just a bit. Anyway, you have your primer here, and this is just showing, okay, it's adding the bases, and then it's imaging as it goes along. Then it goes for a certain distance, say 250 bases, that's read 1. Then it would, for instance, go and start picking up the index, the first index, say the there's actually names for those 2 adapters, the left and the right. One's called the I5 and one's called the I7 adapter. And so you have the I5 index and the I7 index. Okay, so basically in a separate read, you would just a little short read might be just 8 bases long, and you would collect the data that would give you sample interest, like which sample was this? And then this is just showing how read 2 would go. And then you could collect actually the I5 index as well with an additional read, which is what we do in WideSeq. And anyway, so this is just showing basically how it would work on the surface of the flow cell of the instrument. And then just to correct the previous oversimplification, because I was talking about, the Illumina sequencer sequences library molecules. Well, it's not quite that sensitive. It actually sequences 1 micrometer clusters, well, approximately 1 micrometer clusters of Illumina amplicons. And those clusters are scattered over the surface of a flow cell. And you know, each cluster actually derives from a single Illumina library molecule copied about 1,000 times using bridge PCR. So, and this is the promised picture of a flow cell. This actually is not a MiSeq flow cell. This is an earlier type of, well, this is not an earlier, this instrument was basically the big brother to the MiSeq, which we're still using. It is an instrument called the HiSeq, which is now obsolete. I don't think anybody runs them anymore, but this was an extremely popular version of it. And this is showing you a flow cell happened in the HiSeq. In one of its manifestations had 8 different lanes. You can kind of see what it is. Just basically two pieces of glass, and there's like 8 holes drilled into it, and you can flow liquid in there. And you go and lay that on the surface of Peltier junction. You can control temperature as well as image through the clear areas. And so that's basically the main trick behind having this. Okay, but what's on the surface of the flow cell? Well, what's on it are complementary sequences shown in purple and in pink in this diagram to your library molecules. So you're going to denature, you'll see again our library molecules here with adapters on the ends, showing them in pink and the purple. And then you have an insert in gray, and then you're going to denature those, flow them across the surface of the flow cell. And then not shown on this picture is they would actually have to anneal to one of these flow cell oligos. A polymerase needs to extend that and to create a tethered version of that single strand. And then for the next cycle of amplification, it's actually going to, its primer is on the surface of this flow cell attached to the flow cell, and it's going to extend that one. Then when you denature these 2, you'll actually get 2 complementary strands sitting next to each other in opposite orientation. And then if you continue that through several different cycles, you'll end up with clusters of them. 2 becomes 4, 4 becomes 8, so on until, you know, after about 10 cycles, you might have like, say, 1,000 of them in a cluster of the surface of a flow cell. Remember, this is very small scale. It's DNA, so you can fit a lot of them on the surface of a flow cell, and that's the big trick with Illumina sequencing. That's why you get so much sequence. Okay, so we use Illumina sequence to generate WideSeq. So what kind of use cases might you have for that? In general, I generally think about anything up to about, say, 100 kb of template. Double-stranded DNA needs to be double-stranded DNA for that transposase to work on it, will generate some useful results for you. So if you're pushing 100 kb or going beyond that, then you're going to have to expect your downstream results to be more fragmented. And if you're comfortable with that, it's fine. We actually do have customers that will submit bacterial genomic DNA to us, and we'll go ahead and sequence that for them. And it's not like they're going to get a final full length sequence, a full complete sequence of a bacterial genome, but you'll get a lot of sequence from the genome just at a low coverage. And that's still useful to some people. But our major thrust here would be for plasmid. That's what Clint Chappell asked for. I think of it as being especially useful for low copy plasmids, because those used to cause me the most pain with Sanger sequencing. And a lot of times constructs from expression vectors, those are low copy. You're just not getting, it's not like pgem or something or pBlueScript where you're getting tons and tons of DNA from just like a 1 mil. So it's kind of handy in this case because of the methodology we're deploying. We don't really need an enormous amount. Rule of thumb, if you have something that's 1 kb long and you have a nanogram of it, you actually have 1 billion copies of it. So there's lots of room at the bottom here, tons and tons of molecules to work with. Sanger sequencing just happens to be extremely hungry for that. So it's not uncommon for them to ask for microgram amounts of your plasmid if you're doing Sanger sequencing. PCR products are also very popular. You can do long PCR products. You could do pools of PCR products. If you're going to do that, make sure that each product is if it's a mixture of different products, they need to be different loci. If they're the same locus, you're just going to have a mixture of a whole bunch of different sequences. So trying to do a de novo assembly of something in that case would be difficult, and might not yield anything useful. But I think in many cases you might have several different loci you want to monitor all at the same time, so you could pool them all together in the same. You do have to be careful to try to get them in approximately the same ratio when you're adding them to the tube. Two-fold difference is going to make any difference, but it's super easy to be off 100-fold, and then one of your products ends up hogging up all the reads, and you don't get sequence from the other ones. If you happen to have some methylated DNA and you wanted to assay like percentage methylation, you could do typical bisulfite treatment with it and do PCR from that, and then just submit those samples for an assay where we run it through a pipeline called Bismarck to assay basically the amount of methylation you have. And then phage and BACs could work, bacterial artificial chromosomes. Just a word of warning, if you're doing a BAC prep, you really want to use some sort of method that's going to get rid of the contaminated E. coli genomic DNA, to the extent you can. It doesn't matter if it's like 10% or something, but I've seen cases where especially with BACs really are very close to single copy. So they're the same molarity as the host chromosome, but they're like 100, 200 kb, whereas the E. coli chromosome is 4 megabases. So you just end up with tons and tons of E. coli sequence, and you end up wasting it on that instead of being able to use it for your BAC. So there's various methods. Like there's a kit from QIAGEN called plasmid. Well, there's a kit from, I don't know, there may be other manufacturers. I know QIAGEN has plasmid safe DNase, where it basically is an exonuclease, so it won't digest your circular DNA, but it'll digest other linear stuff, which the chromosome will always break into linear stuff unless, well, I won't even add unless, pretty much that's going to happen. Or there's something called a double acetate precipitation, which I've heard used. Basically just a ways to decrease the E. coli contamination. And then, anything else you might think of there, and there have been other things that have been done over the years, basically somewhere between $19 to $50. And I guess at this point I can specify here. So if you're at Purdue, if you're at Purdue and the Purdue Cancer Center, or in the IU Simon Cancer Center, then you get that $19 price. If you're at Purdue, you get a $21 price, and then other people are closer to like $50, unfortunately. But still, $50, pretty good deal, I think. And we do have external customers coming to us for doing this sort of sequencing. Okay, so we do include the first pass informatics along with it, using this pipeline that Rick wrote. And so the idea is, I mean, this isn't just a single sequence read, so maybe thousands of them might be more than you want to page through. And if you don't feel comfortable doing that, we have built in kind of the most common thing to do is like basically a de novo assembly of a subset of the reads. Basically a subset, because in many cases, if you use all of the reads we produce, you get a worse assembly than if we just use parts of them. So generally, we'll try to keep it down, cap it somewhere around like 500x, and we get a better de novo assembly than if we try cranking it all the way up and use everything that we get. I'll actually try a number of those. If it doesn't work, if I don't get a single contig for your plasmid that represents the full sequence, but sometimes it just doesn't help. And, there's areas that we're missing, or the assembly engine is confused because you have some repetitive sequence or some repeated sequence in your plasmid. In those cases, I might email you and say, hey, if you want, I can do a guided sequence if you happen to have a reference sequence. So a lot of times you might have what you expect your sequence to be. You can send me that. And then basically we're more checking like, oh, is it that sequence or not? Anyway, in either of those two cases, we take all of the reads and map them back to the assembly. And the results are stored in a BAM file for analysis with a BAM viewer, such as IGV, if you would like to take a look at them. And then PCR products, another popular one. If you have SNP or SNV calling that you need done, just send me a reference sequence through the email, and we'll map it and use like a caller to call their variations for you. And then you'll get a VCF file back, or depending, there's some other more specialized things where it's a slightly different format if that would be helpful for you. And then, yeah, as I mentioned before, you might, if it's a bisulfite treated genomic DNA that you did PCR on, then we could use Bismarck. Okay, so if you're convinced you want to submit samples to us, you can submit those samples via iLabs. This is the actual, if you want to go directly to our core, you could go directly. I mean, we're 4386 Service Center 4386, or you can search for us in iLabs if you know how to do that, or send me an email and I can send you the link. So after creating the initial, I just want to add this because this, after you do it the first time, you won't even think about it, but it's confusing if you haven't ever done it. After creating the initial request in iLabs, you need to fill in an additional web page via the external URL link that's next to your temporary service ID. You just click on that one and it actually takes you to our web page, because there's information we need that we just couldn't get from inside of iLabs. At that point, each of your samples will be assigned an accession number. So, say you had 10 samples you wanted to run, then each of them would have an accession number. Just go ahead and get some 1.5 microliter microfuge tubes and just write the accession numbers on the top of the tubes, or maybe at least the last 3 digits of the accession numbers. And then add 10 microliters of your sample to the tube, and then put all those samples in a plastic bag with request number written on it. You'll be assigned a request number also. And then if you're here at Purdue, as I mentioned before, you can actually put that into our submission freezer, which is in the lobby of Bindley. And just make sure that you go to the bottom section of our freezer. And then there's a drawer that's labeled Genomics Core, and there's actually a little box in there for specifically you can stick WideSeq samples in. And if you need to ship samples to us, then you can use this address. or again, send me an email if you have any further questions on that. And again, we have this deadline we set up so that say you want to have some sequencing done, you just want to have an idea of like when you're gonna get your results back. Well, if you can get your samples to us by 2 p.m. Tuesday, the 20th of September, then you'd get your results back probably in about a week. Usually takes a little bit less than a week or a little bit more than a week, maybe by 1 day. And then if you miss that deadline, we have them every 2 weeks. Except over winter break, we skip one of those weeks. So basically we have 25 submission deadlines during the year. Now you don't have to wait for the deadline. You can just submit it whenever you feel like it, and it'll go into the next batch. This is just to kind of give you an idea of when you're going to get your results back. And I'll just end here with information about how to contact me. I actually have a listserv. Actually, you will be subscribed to it after a while, because all I do is I almost always am just sending out every 2 weeks. Hey, next Tuesday is the date for the next WideSeq, and I might on occasion put a little bit of extra information in there. And then this is our website. So thank you for listening if you did, and I'm willing to attempt to answer any questions you might have.
All right, thanks, Philip. I didn't see any questions in the chat, but if anybody's got questions for Philip, feel free to unmute yourself and ask away.
Hi, this is Nuria Morral. I have a question. Can you clarify if the price will be including also like the post-sequencing, all the alignment and all the,
yes,
informatics that's involved in, giving us a sequence?
Yes. So it would include those informatics that I mentioned on the slide before. Of course, it's possible to come up with something far more complex. You can do additional downstream analyses that might be beyond the scope of what we would be willing to do for you. But basically, doing the assembly of your sequence and hopefully getting a single. I mean, the goal is to get a single sequence that will represent your entire plasmid in that example, or something similar to that. But yeah, you get all the rest of the stuff happens. You get the BAM files, you get those various different types of analyses. Yes, that's included in the price.
Thanks.
So Phil, you mentioned about 10 microliter of DNA. So what's the quality do you need?
So I would do an actual plasmid prep, and it'd be better if you did. So are we talking plasmid DNA? Is that the idea here? Or
long PCR?
Long PCR. So the quality should be basically whatever you give us, you're going to get the sequence of that back. So if you do long PCR and you ran it on a gel and you see a big giant primer dimer on the bottom of it, you're going to get a ton of primer dimer sequence. So it's a good idea to do some sort of like cleanup. And there's lots of like PCR cleanup kits that'll get rid of most of the, primer dimers. If you have just like a couple of bands there and you're like worried, well, I mean, there is reason to be worried. Say you see instead of one nice crisp band, you see like two bands, then if those two bands are unrelated to each other, then you'll just get the sequence of both of them. And if in general, that would be the way it would work ideally. And, often I think it would work that way. If instead they're you have two bands because you're actually amplifying two different regions of the genome that just happen to be like members of a gene family that differ, say, with the presence or absence of an intron or something, then you might get overlapping sequence. It might be similar to each other, so it might be difficult to assemble that. But as far as I'm concerned, as far as the actual quality of the DNA coming in, usually it works okay. So people, for the most part, if you do like a reasonable cleanup on it, or a plasmid prep, if you're doing plasmids, where you do the plasmid prep, I mean, if you're super old school and you're just doing, breaking the cells open and just precipitating them, you know, you want to do another cleanup step after that of some sort, or at least I would advise you to. You could always give it a shot. I mean, in general, if you're here at Purdue, it's going to cost you $20 to give it a shot. If you're concerned, don't want to spend money for an extra cleanup kit. But for lung PCR, yeah, I would just put it onto a PCR cleanup column and then elute. And as usual, you know, try to avoid. There'll be a wash step and stuff. And the thing that happens with those columns most commonly, if there's going to be a problem, is that you'll end up with like you'll do your wash, and then it'll be a little bit higher than you notice, and a little bit of the wash solution will be touching kind of the nozzle of your column, and the column will wick it. It'll wick back up into the column. And you'll take it out, dump off your sample, and then you'll elute. And actually, when you elute, you'll have a mixture of a little bit of that wash solution, which has like tons of salt in it. Usually has lots of sodium acetate. Will it actually make a difference downstream? Well, we're going to take just some of your DNA and we're going to put it straight into an enzymatic reaction without any other modifications. So it might have an impact. So that said, I mean, a lot of times we'll have like, you know, a run and, you know, all of the samples will produce sequence. So maybe not everything's perfect. You might not get 100% the result you wanted, but you can get it. So it seems to be fairly robust, but yeah, I wouldn't want you to push your luck there.
Philip, I've got a couple of questions in the chat. One, I'll start with, have you sequenced cDNA?
Yes. So there's a couple of different ways you can go with cDNA. Yeah, we've done cDNA. Now, the thing is, you're going to have to actually do double-stranded cDNA, I think. Here's the reason I say I think. I had this strange result where a student from Tim Ratliff's lab, this is years back, took some cDNA they made. This was just single-stranded cDNA, and then they did a PCR amplification on it to get a specific band, and the band popped up and looked okay to them. They submitted the entire DNA sample, the PCR amplification to us, and we got tons and tons of cDNA sequence, just random cDNA. So I don't know what's going on there. The only thing I could think was that somehow the TN5 I would expect it to be very fastidious about whether it's jumping into double-stranded DNA or not, but maybe the heteroduplex was good enough. I don't know. Okay, but so I can't tell you one way or another whether heteroduplex cDNA is going to produce a result or not. You would have to just try it out and see if that works. However, if you actually go to the trouble of making double-stranded cDNA, it should work fine. Yes. Now, if you do that, and you have a very complex pool of cDNA, then you know, you'll get an idea of, say, what the most expressed messages are in your pool. It's not like you're gonna detect information the way that you would from a full, you know, doing a full RNA-seq. I mean, in essence, that's what you're doing. If you take cDNA, you're doing RNA-seq, but you're doing it at very low coverage. It's the same as you know, doing 30 million reads. You're not going to be at that depth. But anyway, yes, cDNA is possible.
So follow up another question. Is there a publication to reference for WideSeq, and would deposition into Geo be required if you want to use this outcome in a publication? I
mean, I would imagine as far as the last question goes, the last part of the question, that depends on that would be the rules of whatever the journal is that you're going to, or the granting agency that's supporting your work. You would need to check with them. So yeah, it's possible. As far as a publication, there have been publications that have mentioned WideSeq there. I have never published a method paper, although my committee is certainly has been encouraging me to do so.
Are there any other last minute questions? All right. Well, in that case, thank you everybody for coming. Thanks, Philip, for for agreeing to give this talk. That was great, very informative. And you know where to reach Philip if you are interested in this technique. Thanks for coming, everybody.
Bye.